Rebirth Consortium is a Marie Curie training network which is a European research platform for scientists. We, as early-stage researchers, study molecular mechanisms associated with brain disorders such as depression and mood disorders. Preventing neurological diseases and the search for drugs to combat them are major scientific challenges. Applications of novel approaches to study diseases affecting mind can reveal new molecular mechanisms in unexpected directions. In the next sections, we will present some of our approaches used in Rebirth Research Consortium. The European Commission funds our work, and training is provided by 2 private companies, 1 non-profit organization, and 7 universities across Europe.
Depressive disorders are a serious challenge in today's society. Molecular mechanisms of these disorders are largely unknown, but generation of new neurons in hippocampus, also called hippocampal neurogenesis, is thought to play a central part in this. In my part of the project, we are searching for biomarkers indicative of hippocampal neurogenesis with proteomics. More specifically, we will compare young rats with plenty of neurogenesis to old ones with less neurogenesis. Hippocampi from these animals will be isolated and fractionated on iodicinal gradient in order to isolate plasma membrane. These will be then separated on STS base cell, and the proteins are digested for mass spectrometer analysis. Proteins found only in young animals will be chosen for antibody generation.
In my part of the project, we generate recombinant antibodies using a technique called phage display. The antibodies created will be used to study hippocampal neurogenesis in living organisms. Studying this can lead to more effective treatments and diagnosis in depressive disorders. Single-chain antibodies are generated in vitro by using phage display method, and they will be used as a tool for identification of neurogenic cells in vivo. How phage display works is, phage library is exposed to immobilized target, which is in this case a candidate we choose from tandem mass spectrometry. Some members of the library will bind to the target through an interaction between the displayed molecule and the target. This selected phage is then replicated in bacteria to get enough material for sequencing. Eventually, the binding abilities and sequencing of the amplified phages are analyzed. Recombinant antibody fragments recently emerged as potential alternatives to monoclonal antibodies. They are less expensive and more genetically stable for therapeutic and diagnostic applications. The antibodies generated in a project can be used to monitor neurogenesis in vivo.
Mood disorder is the most common psychiatric disorder in Europe, especially depression, with a lifetime prevalence of 15 to 20%. It has been reported by the European College of Neuropsychopharmacology and the European Brain Council that depression is already the most important single contributor to the total European disease burden. The major problem with depression is the fact there is no valid clinical signs or symptoms, and in particular no valid biomarker. This project focuses on developing the biomarker diagnostic tool for measuring the neuroactive heptans and their imbalance for the better diagnosis and prognosis in the management of psychiatric disease.
The imbalance between the kynurenine metabolites, the kynurenic acid and quinolinic acid, from the degradation of the tryptophan through the kynurenine pathway has an important neuropsychiatric implication. The core objective of the project is developing robust assay to measure these neuroactive metabolites in tissues and body fluids using the monoclonal antibodies in advanced technology like surface plasmon resonance. On achieving this goal, the produced antibody that is specific to these metabolites are characterized on the specificity, affinity, and sensitivity using SPR. The SPR is a powerful optical detector used in monitoring the label-free biomolecular interactions in real time. The biomolecular interaction between the target metabolite and the antibody immobilized on the sensor surface can be monitored in real time by observing the change in the SPR resonance curve over time. The characterized qualitative monoclonal antibody against these metabolites will be selected in developing the molecular assay kit like ELISA in detecting these neuroactive metabolites in the tissues and body fluids of the depressed patients.